This is the Cresyl Violet project, and we're working in the fall 2020 semester. My name is Abby Manning, and I'm a sophomore biochemistry major. This is my second semester on the project.
I'm Megan Griffin, and I am a junior biomedical sciences major, and this is my second semester on the project.
My name is Jessica Grancelli. I'm a sophomore biology and clinical lab science major, and this is my first semester on the project.
Cresyl Violet is an organic compound used for staining cells and tissue. It's most often used for staining brain tissue and neurons. Cresyl Violet has what's known as a monomer and dimer form. DNA and RNA have bases known as adenine, cytosine, guanine, thymine, and uracil. We will be working with oligos, which are strands of nucleotides, which have the base, phosphate group, and sugar attached. Cresyl Violet forms a complex with DNA and RNA that gives off different colors. These different colors can be seen in DNA and RNA when looked at in a gel. So we have, looking across the gel, we can see that when we have D poly A, D poly G, C, T, and then over on the right side, we have the RNAs. So R poly G, R poly A, R poly C, and R poly U. And you can see clearly that they are different colors. And what we're going to be looking at is these different colors in their spectra using different forms of UV-Vis.
What we've done this semester, we've done research on DNA and RNA oligos with Cresyl Violet. We've done research on DNA and RNA bases with Cresyl Violet, DNA oligos in gel, the pH rate profile of Cresyl Violet, and Peakfit. The UV-Vis spectrometer. This instrument sends waves of ultraviolet light and visible light through cuvettes with the sample. This instrument measures the absorption of the waves through the sample. Using this allows us to analyze the bases and the interaction that they have with Cresyl Violet, and it produces a unique spectrum for each one. Fiber optic spectrometer. This instrument sends waves of ultraviolet light and visible light through gels in which the sample is located. This instrument, it measures the absorption of the waves through the sample, and using this allows us to analyze the bases interaction with Cresyl Violet, and each base produces a unique spectrum, and we can also compare both in-gel spectra with the in-solution spectra. Adding the oligo solutions into the Cresyl Violet solution. We made 0.02% Cresyl Violet solution and diluted it to 0.004%. We started with 2,000 microliters, 0.004% Cresyl Violet solution, and then we added 300 micromolar oligo solution in increments of 10 microliters for the first carry UV-Vis, and then we picked specific amounts to show more evenly spaced out spectra with total molarities of 2.97 molar or 4.43 molar of the oligo.
So this is the spectra we got when we ran D poly C in the Cresyl Violet. So we started with the top line of the spectra is 0.004% Cresyl Violet by itself in the carry UV-Vis. And then we increased the amount of oligo that was in solution to have a total of 2.97 molar D poly C. And as you can see, there's a shift on the left side of the spectra where we think that there's a third species of the Cresyl Violet forming. And then this is D poly A, same thing, where there's a slight shift to the left. This one's just not as noticeable. And D poly G, D poly T, and R poly A, R poly U. And R poly U had also a big shift to the left that was really visible, just like the D poly C did. And R poly G, R poly C. So then after running all the oligos, we wanted to see what would happen if we ran just the bases in solution with the Cresyl violet. So we started by making the 0.02% Cresyl violet solution and diluted it down to 0.002% Cresyl violet. And then we started with 2,000 microliters of that Cresyl violet solution. And then we added 4.5 millimolar of the base solution in increments. So we started with 10 microliters, and then we added either a total of 1,000 or 1,500 microliters. And so then when we ran the spectra, we only ended up doing the DNA bases, and we noticed that it just seemed to be a dilution of the Cresyl violet, and nothing was really happening between the bases and the Cresyl violet.
So earlier in the presentation, we talked about how we can see the different colors when DNA and RNA are stained with Cresyl violet in a gel. So we wanted to use gel electrophoresis to replicate these gels and make another analysis on them using another form of UV-Vis. So gel electrophoresis is a method of separating DNA, RNA, or proteins by their molecular size. It uses an electric field and has a positive end and a negative end. In this case, the top has a negative side and the bottom is the positive side. DNA is negative, so it'll move towards the bottom of the gel when run in the electric field. So the top picture, you can see what I'm referring to when I say the top is negative and the bottom is positive. You insert the samples and the loading die in the numbered wells right at the top. Then you can see our power supply that's run at 180 volts for about a half an hour to 45 minutes, and it runs down the gel. And then I also included a picture of prepping the samples. We heated the samples so that the bands are more clear, and specifically for D-poly G, you can kind of see multiple bands that I'll show in a moment on the gels, but this helps make the bottom band more clear. So this is after the gel has been ran. You can see the loading die has moved all the way down. What you can't see right now are the samples because they are not stained at the moment. We cut the gel at a specific part. I cut the top left corner, so that we can see the orientation, which is written down in a journal or book so that you know which samples are in which wells. Then we stain with Cresyl violet for about a half an hour. And the second picture shows you can start to see the samples. And then we destain. And the bottom picture is after it's destained, and we also have to cut the gel so that it can be placed on a microscope slide so that it can be analyzed under the fiber optic. Here are the labeled samples. So we have D-poly T, D-poly A, D-poly C, and D-poly G. As you can see kind of at the top a little bit, there's some less clear bands, but we're more focused on the one at the bottom. So you can see that they're all different colors as we've referred to before, and we want to get the spectra of these. So using the fiber optic spectrometer and ocean view, we can create graphs that show you the absorbance over the wavelength of each of these. Again, they're all different spectra, but how do these compare to what we did in solution versus this is looking at the in gel? You can actually see from the DNA that we've done is that if you look at the bottom lines on our in solution data, they actually correspond pretty well to what we see in gel. Can kind of match up. So D-poly C is this green, and the shift kind of matches up with the wavelength seen. Now we don't know the concentration of the in gel, but we do know the concentrations of the in solution. So in the future, we'll be starting to look at and compare those and try to figure out the concentration in gel.
So we decided to make a pH rate profile for Cresyl Violet. And Cresyl Violet's pKa's are 7.91 and 10.77. And we think that the double bonded amine is the group that is being deprotonated as we increase the pH. So we wanted to use buffers that are above, below, and in between the two pKa's. So we settled on pHs of 5.92, 7.91, 9.34, 10.87, and 12.74. We were also curious about how the concentration of the Cresyl Violet would affect the spectra, so we chose to do different concentrations of 10 micromolar Cresyl Violet in the buffer, 65 micromolar, and 125 micromolar Cresyl Violet. So this is the spectra we got when we ran 10 micromolar of the Cresyl Violet in each pH through Carey. The top line is 10 micromolars Cresyl Violet, and the line that's pointed to is 10 micromolar Cresyl Violet when it's at pH 12.74. The peaks were kind of small on the 10 micromolar, but there is still that visible shift in the spectra at both pH 10.87 and the pH of 12.74. So we increased the concentration of the Cresyl Violet to 65 micromolar, and the shifts were much more pronounced. And again, when we increased the concentration to 125 micromolar, the shift is still, present and even more pronounced than it was when it was 65 micromolar. So the two bumps on the lower pH are the monomer and dimer forms. So where, the top line is 125 micromolar Cresyl Violet. That's just the Cresyl Violet by itself. So there's two small peaks, and those are the monomer and dimer. And then when you move and look down and look at the 10.87 pH and the 12.74 pH, you can see that there's two very distinct peaks, and they're both about equal intensity. So we think that the reason there are those two peaks is there's like 50% of the protonated Cresyl Violet and 50% of the deprotonated Cresyl Violet, since it's right about around the pKa, the second pKa of Cresyl Violet. And then when we get to the pH is 12.74, there's that third species that the arrow is pointing at that we're not quite sure what that is yet.
Peakfit is a computer analysis program that we use in order to find hidden peaks that are not shown in the data stream. We are using Peakfit to differentiate between the monomer and the dimer forms, and we would like to see what percentage of each form is present. The x-axis is wavelength, and the y-axis is absorbance. So this is the Peakfit analysis for 50 microliters of D-poly G. The peaks are seen at 589.95 and 500.73. And we only use the values between 400 and 700 for all of the runs, because that is where the peaks for the monomer and dimer forms appear. The taller peak is for the monomer form, and the smaller peak is for the dimer form. This is for 2 microliters D-poly A. The dotted line is the original peak that we got from our spectra, and then the tall peak, which is labeled 583.65, is the monomer form, and the smaller peak, which is 525.99, is the dimer form. And then I compared different concentrations of D-poly C to each other to see how the monomer and dimer forms would change. And for the 10 microliter D-poly C, the monomer and dimer forms were closer in amount to each other compared to the 2 microliter D-poly C, where the dimer form was significantly smaller for that peak. And then for 2 microliter D-poly A, I tried to find percentages for each peak. So the first graph shows where the peaks are located, and the second graph shows what percentage of the overall peak that they are showing. So the monomer form is 87% and the dimer form is 22%.
Moving forward, since the bases seem to be just dilutions, we're going to be moving away from those and focusing more on the oligos for DNA and RNA with Cresyl violet. We still have to do the gel electrophoresis of the RNA oligos because this semester we only got the DNA ones done. We are also going to order another form of Cresyl violet and create a pH rate profile for that. We are going to continue using Peakfit on all of our spectra and give us a better data analysis. We want to continue using Peakfit and better understand and interpret all of our data.
We also want to acknowledge and thank Dr. Charleboy, Jane Schubert, the Nazareth College Chemistry and Biochemistry Department, and the former research students who have worked on this project. Without them, we would not have been able to advance the project as much as we have this semester.